Good afternoon, colleagues. Let me share with you the outcome of international studies on catalytic antibodies endowing with specific activity along with antigen binding capacity. Unfortunately, to personality, this is genius, greatest one, number one in the world in autoimmunity. He died last year, COVID, 77 years old. Director of the Center of Autoimmunity at Johns Hopkins and head chair of the Department of Autoimmunity at Harvard Medical School, biotech. So at this point, only two personalities, very much and me, are keeping on the studies. New targeted therapies for autoimmune and chronic inflammatory disorders would require greater understanding of a patient and now a pre-illness person at risk to get the therapy personalized for either of the above-mentioned subsets. In this sense, the impact and implementation of diagnostic, canonical, predictive and prognostic, the upgraded ones, biomarkers, becomes the holy grail of platforms, algorithms, and protocols, which are the crucial for personalized and precision medicine as the latest and fully upgraded model of healthcare services. Just the definition made by NIH regarding biomarker, it's not so attractive already. Biomarkers enable pre-early, in precise term, subclinical diagnosis, guide targeted therapy, and monitor the activity and therapeutic responses across the diseases. Strategies for discovering biomarkers, global strategies, are depicted here. So it's necessary to identify biomarkers of the next step, and again, next step generations, and to create a fundamentally new strategy based upon subclinical and predictive recognition of those biomarkers long before the disease clinically manifests itself. Among the best validated predictive biomarkers today are autoimmunity-related ones to predict and to prognosticate risks of the chronification, complications, and thus disabling. Look, it's just predictive antibodies as biomarkers to monitor chronic autoimmune diseases. It's just a gift of my very close friend from Tel Aviv University, Yehuda Shonfeld. Autoantibodies as biopredictors of specific disease manifestations in specific autoimmune, systemic autoimmune condition. Look. It's new, but already old. Okay. Since chronic autoimmune disorders are preceded by a long subclinical, usually symptom-free phase, The latter is so much valuable and important since chronic autoimmune inflammation course is structured to consist from different stages, including subclinical and clinical illness. Meanwhile, the utility of predictive biomarkers to monitor and to manage chronic autoimmune inflammation and to predict stepwise transitions is dependent on three key parameters, which must be carefully, in precise manner, assessed: sensitivity, specificity, and positive predictive or prognosticable values. Why biomarkers failed at this point to be implemented into the clinic? There are lots of problems, which is illustrated here. So you might understand that accurate prediction is crucially vital for prevention of autoaggression, and the targeted treatment could thus be given to those individuals who are most likely to develop a disorder. For example, might be somebody so do not know, but it's widely known nosology, multiple sclerosis, MS, is just one of the chronic autoimmune diseases resulting in the destruction of myelin by different tools, including autoantibodies. The crucial step in the pathogenesis of MS is a primary myelin damage, which is mediated by autoantibodies to trigger a release of separate and pathogenically valuable myelin-associated epitopes into the bloodstream and later to attack myelin. Those epitopes act as sensitizing factors to generate autoantibodies more and more, which in turn will drive the demyelination and thus the disease progression. A spectrum of myelin-associated autoantibodies occurring in MS patients has been confirmed to be very large. Look, just some of them. MOD, MBT, and 10, 10, and 10. Keep in mind, crucial one, MBT, myelin basic protein. Along with canonical antibodies, some of the families proven to occur are antibodies in those, but I would prefer the term armed with catalytic activity, catalytic antibodies or enzymes, antibody, enzyme. Professor Paul, 1975, has died, Stanford University. And thus to belong to antibodies with a feature of functionality, more precise, B functionality. Look, antibody binding specificity and enzymes catalytic activity being induced, and thus functionality. Catalytic antibodies or enzymes are multivalent immunoglobulins, presumably of IgG isotype, endowed with capacity to hydrolyze the antigenic substance. This property match is very is AB fragment. Why it's not seen? Blue. Of the Ig molecule and is appearing to sound as a functional property of the antibody molecule as a whole. In this sense, proteolytic antibodies or antibody-proteases as a significant portion of the big family of enzymes represent antibodies endowed with the capacity to provide targeted proteolytic effect. The examples of the mention. Ig found in a series of autoimmune disorders to cleave targeted autoantigens and thus to act as informative biomarkers to monitor both clinical and subclinical causes of the disorder. Due to the London Institute of Economy compensating subclinical stage in chronic disease, it's 62% of the investments in the disease as a whole, not for physicians who are curing clinical manifestations, not so valuable already. For instance, in bronchial asthma, to cleave vasointestinal peptide VIP as the major targeted autoantigen. Could you read it yourself? Or not visible? This is the first catalytic antibody. The award of Rockefeller in 1979. The other example is a family of antibody-proteases detectable in autoimmune myocarditis to cleave cardiac myosin. It has been done in my laboratory in a tandem with Harvard Medical School and Johns Hopkins, but he has already told you that he has just died last year, not else. And in autoimmune thyroiditis, to cleave thyroid autoantigens as the major autoantigens and thus informative biomarkers to monitor the causes of autoimmune thyroiditis. The same. A situation of greatest unique interest is occurred in multiple sclerosis, MS, which is a chronic autoimmune inflammation in central nervous system, CNS, and resulting in demyelination, axon loss, and development of phenomenal disability. The latter mentioned would further demand the stage of remyelination, including regenerative manipulation of the newest generation using the proteolytic property of the antibodies. Meanwhile, the likelihood of autoantibodies and biochemical elements of multiple sclerosis has been proven to be proportional to the presence of anti-myelin autoantibodies and anti-MBP, myelin-based protein, as I already stressed the value in particular. Anti-MBP antibodies dominate in MS patients to accompany their myelination. We have established that anti-MBP antibodies harvested from MS patients and mice with MS and analog experimental autoimmune encephalomyelitis exhibited MBP-specific proteolytic cleavage of the MBP molecule, not MAG and MOG, not other myeloid-associated molecules. It's one of the first publications regarding this. The activity of antibody-proteases markedly differs between MS patients and healthy controls. I guess you are seeing it. And in patients with different causes of MS, I would just let you know globally, 2 causes, not 2, but 2 globally. Aggressive and moderate. You see the activity of antibody-proteases drastically different. Drastically. Okay. And finally, the activity demonstrated also significant correlation with scales of demyelination, neurological deficiency, and thus with the disability of the patients, as it seems from the EDSS Kursky clinical scale. You see. Of the great value is sequence specificity of antibody-proteases, which was analyzed by a complex of the modern technologies. Look, antibody-mediated proteolysis of MBP results in generating a set of peptides with molecular masses ranged in various but fixed boundaries to assume common principles of molecular kinetics of MBP. The final statistic data revealed 5 sites of preferential proteolysis of MBP autologous, MBP by autologous antibody-proteases. Most of those sites, as I said, defined, are located within 43170 region of MBP, which are just immunodominant. 2 sites from the site located within 43170 sequence would comprise 81103 and 8298 subsequences that, while being sequence-specific, highly immunogenic and encephalitogen, both proved to be specific and major inducers of very aggressive experimental MS in mice. The most immunogenic and encephalitogen sites of immunodominant category responsible for generating aggressive bursts of demyelination are concentrated in 3 areas of MBP molecules. Strongest one, smallest one, 8298. A weaker one, in longer, 81103. A site with the lowest immunogenic and encephalitogen properties is formed by a rather long sequence defined. The final step of the study is concerned the evolution of antibody-associated proteolytic activity at different stages of the disease progression. For those purposes, we have been monitoring MS patients, their direct relatives, and healthy volunteers for around 4 years. Look, MS patient, relatives, healthy controls, drastically different. The relatives being seropositive for the antibody-proteases were being monitored for three years, whilst demonstrating a stable growth of MBP-targeted antibody-associated proteolytic activity. Please, read yourself. As the disease progresses to transform from subclinical symptom-free into clinical stages, the activity of antibody-proteases is being escalated to reach the indices we have established from as patients with a diagnosis of clinical illness concerns. As it's known, canonical antibodies play neither predictive nor discriminative role to affect the subclinical stage of MS. Meanwhile, sequence-specific antibody-proteases have proved to be greatly informative and thus valuable as biomarkers to monitor MS at both subclinical and clinical stages. Therefore, the proposed predictive value of MBP-targeted antibody-proteases for the development of MS is being challenged. The traditional goal of antibody engineering is to combine various antibody domains to generate customized antibodies that show specialized binding properties, optimal half-lives, and desirable effective functions. Antibodies can be engineered to make proteins of high affinity or smaller molecular variants that retain or change the functional properties of the original antibodies. The last years have seen a major upturn in the fortune of therapeutic antibodies approved for clinical use, including chimeric, humanized, and monoclonal antibodies. Emerging have created a vast range of novel antibody-based therapeutics which specifically target biomarkers of disease. Antibody-protease engineering in this sense would offer the new upgraded ability to enhance or alter the sequence-specific activity to expand the clinical utility of the absolutely new translational tools. So, in this sense, antibody-proteases can be programmed and reprogrammed. We have already done it, jointly set with Osaka University in Japan and with the UCSF in San Francisco. It's just the first step, and now we are looking and keeping on negotiations with companies regarding production of targeted antibody-mediated translational tools. No time at all, so I understand this. We are now experiencing a renaissance, primarily driven by next generation biotechnology. So I guess that you would understand that personalized and precision neurology would dominate in the narrow field of clinical practice very, very soon. Thank you so much for your patience.